Okay, back to complete our meiosis discussion. Meiosis is an interesting part in the semester. I like this part because, again, we learn how we make the gametes, we learn some differences, but I hope you understand when we get to meiosis, this is the basis, the precursor for genetics. And genetics is the transmission of DNA from parent to offspring. So to understand genetics, we need to understand first DNA, the macromolecule DNA. We understand how DNA can replicate itself. We understand the cell cycle, mitosis, this new cell cycle, meiosis. And then we start to get into particular instructions on the DNA, chromosomes as well. So we begin our discussion now on the idea of sex development. So sex chromosomes determine the sex of the child. So we've talked about this a little bit. We say that the female sex is the default sex. When we talk about steroids early in the semester, lipids, we know that steroids give a signal to increase testosterone, and then we start to develop into the male trajectory. So how or what sex a child develops into is dependent first upon DNA, and then secondly upon the appropriate timing and appropriate amount of steroid signaling, right? So when we come back to our genetic basis here, we know that in the female will receive an X instruction from mom, an X instruction from dad. The male will receive an X chromosome and a Y chromosome from the dad, thereby generating a male. So what is the distinction between these two sets of chromosomes? The X chromosome is going to be a longer chromosome. It's got more genetic instructions, more genes. The Y is a much shorter chromosome with much fewer instructions. So literally speaking, right, ladies, you are more genetically complex than the males. You have more genetic instructions. We don't have all the genetic instructions you have on the X chromosome, right? We have one X chromosome, but we also have the Y. But on the Y chromosome, there's a gene, an important instruction that determines what we call maleness. It triggers then the steroid signals to occur, forms the external genitalia, and basically makes the male anatomy. So again, ladies, you're like the beer, right? Dos X, right? Two X's. So you have two X chromosomes, males, the X and the Y. So this is your somatic condition, diploid condition. So ladies, what can you contribute to your child? Well, you can contribute an X or an X. That's all you have. You can send one X or the other X, but ladies only contribute the X chromosome to their offspring. Males, their diploid condition is XY. In meiosis, they can send the X or they can send the Y chromosome, right? So with that being said, now we know that is the male, is the dad that determines whether or not the child will become male or female, depending on what the male contributes. We know ladies are going to contribute the X. If a male contributes an X, we end up with two X's, female. If the male contributes the Y, we end up with XY, male. So it's, again, ladies, all you can pass down is the X. So the male determines the sex of the child, whether he passes down the Y chromosome to make the male or the X to make a female. A lot of words here, right? But this gene, this SRY gene on the Y chromosome, right? So that is the sex determining region on the Y chromosome, SRY, sex determining region on the Y chromosome. The maleness determining gene. So this gene codes for a functional protein that allows testes to develop. So if we don't have this SRY gene, we don't allow testes to develop. There's no testosterone, so ovaries will develop instead. So the Y chromosome has the instruction that allows testes to form, which allows the male anatomy to develop. On the X chromosome, right, there is what we call the DAX1, right? Ooh, this is not nice for the males, right? This is called the anti-testes factor. So the X doesn't want testes. The X's job is to generate an ovary, a female. So the Y gene comes in, and here we get into a concept. I don't know if you're aware of this concept or heard of this phrase. Is they say sometimes it's easier to ask for forgiveness than for permission, right? So if the Y chromosome asked for permission, yes, excuse me, X, can I make testes? What is the X going to say? Say, no, 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 no. This DAX1 instruction on the X chromosome says there will be no testes here, right? No testes, right? So what the Y does, because it's a smaller chromosome, it can transcribe, remember these terms, transcribe and translate and generate those proteins a lot faster. So it does its job. It produces the protein, the testes develop, and then we have the male. So we get into the idea now of recessive genes and dominant genes. And this is, again, built into the concept of genetics. So the Y chromosome, the Y instruction, the SRY gene basically transcribes and translates faster. So it is a more dominant gene than is the DAX1. So if we have two X chromosomes, again, anti-testes, no testes, right? Here we have anti-testes and testes. So who's going to win the battle? Well, who is going to get the protein generated fastest? And that's the Y. So again, the role of the SRY gene is to actually inhibit the maleness inhibitor encoded by the DAX1. So by having this Y chromosome making this protein, we get the correct genes going, we get the correct steroid signals, and it's, again, each Y chromosome can compensate for one X chromosome. That's going to become significant in a bit. So I hope all that made sense. Just introducing our chromosomes, our dominant recessives, build into the genetic sort of concept here. All right, so let's say mom is pregnant, and early on in the pregnancy, there's some complications. They take her in. There's a couple of tests they can do, right? We have, early on, we have this what we call chorionic villi sampling, that we can take some, basically some cells from the chorion that's helping to develop the placenta, the amniotic sac. Those can provide cells. A little bit later in development, it can stick a large needle, extract cells from around the fetus, from within the amniotic sac. Regardless of how it's acquired, once we have these cells can then be centrifuged, and then those cells can be broken apart, and we can then extract all of the chromosomes. And this process is called doing a karyotype or karyotyping. So karyotyping, again, requires us to extract fetal cells in multiple ways possible. Those cells then, we get the cells, the cytoplasm, the cell is ruptured, the nucleus is ruptured, and we then extract all of the chromosomes. The chromosomes are then paired, homologous ones, the one from mom, one from dad, one from mom, one from dad, one from mom, one from dad. And this is what a unsorted karyotype looks like. It's hard to work with. Luckily, with computers and scanning, this can be done now with one press of a button. We can arrange our karyotype something like this, color-coded and size-coded, and this gives us a lot of information now, right? This karyotype. So what we're looking at now is the chromosomes found in each and every cell of this unborn fetus, right? And we can do a lot of genetic analysis from here, right? One, we look for our magical number of 46. Do we have a diploid condition of 46? So we have here our 22nd pair, our 23rd pair, which is the X and the Y. I see a match on each chromosome. So yes, our 46 number is valid. That's good. There's nothing missing. There's nothing extra. And I can determine at this point if it is a male or female. And I see where there's an X, oh, and there's our little Y. So I would know then that this is a genetically healthy, no genetic abnormalities, and male fetus that's developing here, right? So a lot of genetic information, a lot of important health information can be derived from this karyotype, right? And it's not necessarily that invasive. I mean, I say that, but I'm not the one getting a big needle injected into me. But compared to other tests, it's really not that necessarily invasive. And we can extract a lot of valuable information from this karyotype. All right, so not as colorful, right? But here we have a normal female karyotype. How do we know it's a normal female? We have an X and an X and no Y. So DAX1, DAX1, anti-testes, no Y chromosome. And everything looks good, everything looks healthy. Again, a normal male. Why male? Because we have the X and the Y. And this is just standard. They always start with the longest chromosomes, chromosome 1, and then they get to the shortest chromosomes, chromosome pair 22. And then the X and Y typically are always at the end, right? So at the end down here, or at the bottom down here. Okay, so this is what's supposed to happen. And this normally does happen, but since we're going into the medical field, we know that sometimes there's things that go wrong. And one of the main problems is what we call a non-disjunction error. So non-disjunction, to not separate correctly. That's what that translates to. So a non-disjunction error, the chromosomes do not separate correctly. So this is a meiotic error. It can happen in anaphase 1 or anaphase 2. So we're supposed to start with a diploid cell and end up with a haploid cell. Start with 46, we're supposed to end up with 23 in each gametic cell. Well, sometimes that doesn't happen. We have a non-disjunction, which generates an aneuploid cell. An aneuploid is an abnormal number of chromosomes. This is caused by, again, a non-disjunction. And when we have these aneuploids, we typically have the loss of the child, a miscarriage, or some sort of birth defect that develops. So again, we don't want these to happen. They do happen in humans, but if we can avoid them in some way, we do want to avoid these to ensure that the chromosome number will be appropriate in the fetus. So here we see meiosis 1, anaphase 1. So we're supposed to send, we have a total of 4 just for this example. So 2 and 2, so we're supposed to send 2 to this side and 2 to this side, right? So this side worked. Anaphase 1 worked properly here. Anaphase 1 didn't work properly here. So now we end up with 3 chromosomes in one cell and only 1 in the other. So, well, meiosis 2 works properly. And we end up with 3, 3, 1, and 1. We should have had 2, 2, 2, and 2. So we have haploid plus an extra, haploid plus an extra, haploid minus 1, haploid missing 1 here. So if the non-disjunction error happens in the first anaphase in meiosis 1, none of the gametes will be regular or normal. If the non-disjunction happens in meiosis 2, in anaphase 2, well, we still have a chance, a 50% chance that, this won't work normally, that 2 out of the 4 gametes will be normal and happy and healthy, right? 2 of the 4 will be aneuploids. We have 1 with too many, we have 1 with not enough. Now, this becomes a statistical chance, right? If these are sperm cells, let's say these are all sperm cells, which of these cells is going to be the one that's going to go fertilize the egg, right? So if it's one of these 2, great, that's fine, we're set. If it's one of these 2, then again, it's not going to contribute enough DNA or too much DNA. If it's any of these, all of those are going to be abnormal there. Right? So it becomes a statistical chance as to which of those sperm cells then ends up fertilizing. Same thing for the female. Which one of these cells is going to be selected early on to become the egg? Right? Hopefully, statistically, it was a valid one. Right? So again, just there's many types of problems that can occur to produce birth defects. This is of specific type that we call the non-disjunction errors. Okay, so non-disjunctions lead to aneuploid cells, right? Altered number of chromosomes in daughter cells, and this is important. Oh, and let me backtrack here, sorry. So when we talk about chromosomes, right, they're all chromosomes. All of these 46 things here are chromosomes, but we distinguish the X and Y as sex chromosomes and all of the other chromosomes as autosomes. They have nothing to do with sex determination. They form the brain and the heart and the lungs and the kidneys and the body. Autosomes, they form yourself, your auto, right, yourself. The X and Y, they do other jobs as well, but they're involved in sex determination. So X and Y being the sex chromosomes, all of the others being the autosomes. So again, if there is a loss of an autosome, if we end up with something like this, where there's not enough chromosomes, if we don't inherit enough autosomes, that is always a lethal combination. So early in development, not enough chromosomes were developed, and basically, or not enough chromosomes were inherited by the fetus, so the fetus doesn't have enough genetic instructions to form the body properly. So that generates a natural miscarriage, right? It's sad, but it's Mother Nature's way of saying this little combination of chromosomes is not right. So it terminates the pregnancy early on. So we must have all 44 autosomes, right? There's no other option. We have to have 44 autosomes plus then our 2 sex chromosomes. If we have an extra autosome, so we inherited too many autosomes, we have enough instructions to form everything, but now there's an extra set of instructions that complicates or disrupts normal physiology. So an extra autosome is lethal most of the time. So normally it will cause also a miscarriage, but there's exceptions to that rule. Now, additional sex chromosomes have less serious consequences because they're not forming necessarily vital body components, right? They do other important things, but they're more involved with the development of male or female, right? So we have to have the exact number of autosomes to ensure viability. If we don't have enough, that's lethal. If we have too many autosomes, lethal usually. Incorrect number of sex chromosomes, X or Y, we're not sure what effect that may have. We'll get into some of that here. Okay, so mom and dad, nervous. Mom's been having some complications. They take her in, they perform amniocentesis. They extract our cells, perform a karyotype, and this is presented to you. So after analyzing this, what would you say is going on here, right? Is this a genetically normal, a normal non-disjunction type of karyotype? No, it's not, right? So if you notice here, chromosome pair 21 is not a pair. We have 3 chromosomes here. So we call this trisomy 21. 3 chromosomes of the 21st pair here. It's a girl, a female. We have our 2 X's. But we know we have an extra autosome. So this becomes a high-risk pregnancy. The extra autosome is lethal most of the time. So this pregnancy naturally might miscarriage. It may not go full term. If it does go full term, right, or if the parents decide to allow it to go full term, the child is born with a condition that we call Down syndrome. So if you see a Down syndrome child, you know that they are survivors. They beat the odds. Mother Nature basically didn't necessarily, I'm not gonna say didn't want them to survive, but didn't give them the correct genetic composition to successfully enable them to survive. But they beat the odds. They were born, but they're gonna have some deficiencies. If you notice, children with Down syndrome look similar, and it doesn't matter the parents, the nationalities, the ethnicities, none of that. If they have Down syndrome, they all have similar sort of body features, right? They have the short digits, splayed toes, facial features, that extra little fold in the eyelid. They have what we call the common phenotype. They look similar because they all have that genetic similarity. They have that extra 21st chromosome. They have below average IQ. So mental capacity is typically deficient, although there's a range of severities there. They typically have problems with their heart because on chromosome 21, there's some important heart valve instructions that they don't form properly. And we don't have our important 46. We don't have an even number. We have an odd number. So that is going to make it complicated in the normal meiotic process to end up with a correct number in the offspring as well. So chromosome numbers that do not equal to 46 typically have an impact on sterility, or infertility. These children cannot, I'm going to say usually, I'm sure there's been a few weird exceptions, but normally they cannot have children of their own. So Down syndrome, trisomy 21, the product again of a non-disjunction error. And again, very difficult question to ask and or answer, right? I don't think either parent wants to know they were responsible for this, but it can come from either parent. Mom could have been the one that passed down the extra chromosome, or dad could have been the one that passed down the extra chromosome. Now, it can come from either parent, but normally it's associated with the female that passed down the extra chromosome, right? And why does this happen? Well, we got to go back to the age of mom and the age of the actual eggs. So remember, ladies, you had your eggs since birth. So if you're 18, 19, 20 years old right now, that's how old these eggs are. And anything and everything you've exposed your body to, radiation, x-rays, medication, drugs, whatever, all of that has also affected your eggs. For the males too, but males don't, the age of the sperm is much less than the age of the eggs. Males are constantly regenerating sperm, whereas ladies, you have those eggs since birth. So there's a very strong correlation. As the age of mom increases, the risk of Down syndrome increases as well, right? Those eggs been subjected to a lot. The spindles become kind of non-efficient and they don't separate the way they're supposed to. So again, if you look at most Down syndrome children, not all, but again, most, you'll notice that the mom and dad were a little bit older when they had these these Down syndrome children than the young parents here. So just, again, part of the biological facts. May take all that into consideration if you are considering having a family, right? And it's a tough chance. It's a tough sort of sociological fact to deal with, right? If parents are too young, they don't have good jobs, they're not economically stable, it's difficult to raise a child, right? If you postpone having a child till you're older, financially you're better off probably, but biologically, again, risk factors for different things increase as well. So it's always a challenge. And again, you always hope for the best when there is a pregnancy, that you're dealing with there, right? So we carry on. We look at Down syndrome as an autosomal non-disjunction, right? And I'm not going to cover many other autosomal non-disjunctions because there's not very many other autosomal non-disjunctions. There are a few, but remember, we don't have the proper number of autosomes. It's a natural miscarriage, right? Termination of the pregnancy. But we come now to the sex chromosomes. This is a different situation, right? So autosomes are correct. And now we look at this here, right? We look at a situation where we have one X and nothing else. We call this monosomy X, one X chromosome, also known as Turner's syndrome. So diploid condition equals 45. So typically non-fertile. We say this is genetically a female. And why would you say this is a female? There's no X. I should say there's no Y. There is one X, but there's no second X. So why does it continue to the female sex? Again, because there's no SRY, there's no Y gene. So we stay on that default sex developmental pattern of female. Intelligence doesn't seem to be affected, right? So it's basically a normal female, again, quote unquote, normal female, but without that extra X, wrong number of chromosomes, and infertile. There's a famous actress, Jamie Lee Curtis, who actually has monosomy X. He's written some books and talked about her condition, but she's successful. She's healthy in all regards, except again, sterile and missing that X chromosome there. So monosomy X. So as far as genetic conditions go, might be debatable, but again, I would say this is not as severe as the Down syndrome, right? They cannot biologically have their own kids, and for some people that would be very devastating. But again, they're healthy in pretty much all other regards. Okay, how about this one, right? So mom and dad, you're the proud parents of a little baby XXY. What do we have here? XXY, male or female? Hope you said male, right? This is a male because we have the Y. Although the Y is typically used to fighting only one DAX gene, one X chromosome, now it's got to fight off two of them, right? So we have two anti-testis factors. So there's going to be some slight issues here. So Klinefelter's XXY condition will generate a male, right? 47 chromosomes, sterile. We get into some interesting physiological, psychological, sociological conditions. There seems to be average or below average IQ, so there is some impact on the intelligence of the individuals. They tend to be taller, right? Why taller? The hypothesis is they get both a testosterone growth spurt and an estrogen growth spurt. So they typically grow taller than normal. There's some other sort of characteristics. So here's a normal XY male. This is an XXY male, right? So again, they're male. They're not hermaphrodites, or they're not gay necessarily, whatever. This has nothing to do with orientation, sexual orientation. This is only anatomical formation. So if you notice, they're taller, they have narrow shoulders. So kind of, again, related to the two X's. Gynecomastia, they develop kind of like man boobs, breast tissue, small testes because, again, of the anti-testes factor that we're fighting off. The infertility, they have basically the hip structure is going to be more feminine, wider hips, flared out pelvic girdle, fat deposition in areas more feminine rather than more masculine. I would basically wonder what happens during the puberty effect, right? When they start being attracted to different, they're getting the testosterone signal, but they're also getting maybe reduced testosterone or maybe even estrogen signals. So a lot of these individuals basically would struggle with their sexual orientation. They would struggle basically with their identity of self. Are they, maybe they look male, but they feel more female, right? So then we get into some secondary surgical procedures or hormone therapy that can be done for them to achieve their, the body and the mentality that they're comfortable with, right? So psychologically, they are very interesting to analyze. I'm sure they don't see it that way, but again, the Klinefelter syndrome there. This one I found, this is just female, right? But multiple X. So I don't know what's going on. We have a lot of just weird stuff. Mom and dad did not take care of their DNA. Chromosomes are mutated. We have blebbing. We have, again, just too many. There's a two major non-disjunction situation going on there. So this one is different, right? So this is we saw XXY. Here we have XYY. So mom and dad, you're the proud parents of a little boy, a little manly boy, right? Extra testosterone, extra manliness, if you will. Yeah? Again, extra testosterone, again, still sterile, not fertile. We have, again, that extra dose of testosterone, which, again, is going to have their impact on their behavior, their development, aggression, short-tempered. Think of like young boys going through puberty. They get aggressive with the surge of testosterone. So that basically would be the effect for these individuals here. So the moral of the story, take care of your DNA, take care of your chromosomes, because your children will benefit or will suffer from basically the care or lack thereof that you provide in these gametes there. So kind of what we talk about, right? So monosomy X, Turner syndrome, Klinefelter's XXY, the triple X syndrome ladies. We looked at the XYY. It doesn't have an actual name. We just call it XYY syndrome. If we have a monosomy Y, we only receive one Y, no X. There's too many instructions on the X that are missing. That child also becomes sort of miscarried, a natural lethal combination. Okay, so more stuff to cover, but we'll leave it with that for now. I just have to cover twins. That'll be the next discussion, and it'll be the last section on meiosis.